Hello, everyone, and welcome to another episode of Nowhere Yudarris. Today we'll be uncovering biology's own cell theory. We'll be going through the organelles of cells. We'll be going through bacteria and viruses' way of replication and the life cycle of a bacteria. So, we're going to start with the organelles of the cell. This is a eukaryotic cell, and what a eukaryotic cell is that um, it's not a single celled, all right, like a prokaryotic cell, right? It has a lot more organelles. It has a nucleus, and this is where we start talking about the nucleus. The nucleus is where the DNA and RNA are stored. The nucleus is surrounded by a nuclear envelope for separation and translation. Translation as well um, is a significant part of why the nucleus is really significant to each individual cell. And then surrounding the nucleus is the nuclear membrane, which is selectively permeable. What does that mean? It means that it lets in specific things and doesn't let in other things. Alright, then we have the mitochondria. You probably heard about this. Mitochondria is the powerhouse of the cell. Alright, and then inside there are foldings. If you can see a little bit here, the foldings inside the mitochondria are called cristae. What a mitochondria is and what really makes it special is that they're semi-autonomous. So the way they replicate, semi-autonomous meaning that um, they kind of have a semi-system of their own in terms of like replication. So the way they replicate is through binary fission, which we'll be uncovering later on. I'll talk about it later on. What's also special about a mitochondria is the fact that it can trigger and induce apoptosis by triggering the electron transport chain, which I'll be talking about in another episode. Then we have the lysosomes. Lysosomes basically just break down substrates ingested by a process of endocytosis and response against foreign substances. So lysosomes can also act as protectors for the cell. And then we have the endoplasmic reticulum. Alright, endoplasmic reticulum can be a little bit tough for some people. Alright, and the way I want you to remember it is this way. You have the rough endoplasmic reticulum, alright, for ribosome. So I want you to remember this. Rough ribosome, R and R, alright? Rough ER for ribosome for translation. And then you got smooth ER. Smooth ER is basically for lipid synthesis and detoxification of some drugs. Alright, so let's review that again. Rough ER are ribosome translation. Smooth is for lipid synthesis, so smooth synthesis lipid, right? And detoxification of some drugs. Then we have the Golgi apparatus. So the main function of the Golgi apparatus is that it helps process and package proteins that need to be exported out of the cell. So in terms of the protein synthesis process, um, there are four steps, and the fourth step, the last step, is kind of like tying the ribbon. What happens in Golgi apparatus. Then we have something called peroxisomes. Peroxisomes. So they have hydrogen peroxide. The way you can remember it is hydrogen peroxide, peroxisomes. Hydrogen peroxide, peroxisomes. So they break down fatty acid chains and participate in phospholipid synthesis. So peroxisomes, how do you remember its functions? So it has two functions. Alright, so peroxisomes participate in phospholipid synthesis, so PMP again, and they break down long fatty acid chains. Then we're going to come to what a cytoskeleton is. What is a cytoskeleton? A cytoskeleton basically has three structures, and these three structures help with support and help with interactions and connections. So first off, we got microfilaments, microtubules, and intermediate filaments. So microfilaments are made up of actin rods and provide protection and use for muscle contraction interaction with myosin. Alright, so microfilaments, the way you can associate it with it is that it's in the muscle, alright, and it's made of actin rods and provide protection. So microfilaments provide protection used for muscle contraction and associated with the interaction with myosin. Then you got microtubes. Alright, they're hollow polymers of tubulin proteins that provide a primary pathway for motor proteins like kinesin and dynein. Alright, so microtubes, hollow polymers, provide primary structure for kinesin and dynesin. Alright, where can you find microtubes? You can find microtubes in cilia, flagella, or kinocores. So cilia is composed of microtubes for movement. Flagella is the movement for stuff like sperm, and kinocores. So the division where microtubes attach to central cell, that's meiosis and mitosis. So the way to remember microtubes is basically motor proteins and movement like kinesin and dynein, and cilia, flagella, and kinocores are where microtubes can be found. And then lastly, intermediate filaments, and they maintain the cytoskeleton structure such as keratin. Do you know what keratin is? So look at your nails right now. This is keratin. So moving on, we have epithelial tissue. Alright, personally, I even struggled a bit at the beginning of understanding what really is epithelial tissue, but epithelial tissue is involved in protection against pathogens and absorption and secretion, and it's connected by something called basement membrane. Epithelial tissue does a whole lot of stuff. So basically, it's involved in protection, absorption, and secretion. So you can, I guess, call it 3 in 1, 4 in 1, something like that. And then it's connected by basement membrane. Epithelial tissue is one of the tissues, right? You know, since its name is epithelial tissue, but it can also act as an organ. But when it acts as an organ, it's called parenchyma. So a parenchyma polarizes with one side facing the inside of the organ and one side with the vessel. So the way I want you to imagine this is, let me get my pen. So let's say this is a vessel, all right, and let's say this is an organ, okay. This is where the parenchyma would be, right, in between them. I want you to ask yourself, why would that be? Alright, so the reason this epithelial tissue, also called parenchyma, does that is selective permeability. Means that it allows certain stuff and doesn't allow certain stuff, allows for absorption and secretion and all the stuff we just talked about. There are three types of epithelia: simple epithelia, which is one layer, and then stratified epithelia, which is multiple layers, and then pseudostratified epithelia. That's just one layer and appears multiple. Its shapes are cuboidal or columnar or squamous. Alright, moving on. We'll be moving on from the eukaryote a little bit, but not too far. So we got an archaea. Archaea is a single cell organism with genes and metabolic pathways. So archaea is basically a mix between prokaryotic cell and eukaryotic cells. Alright, and they are extremophiles. They thrive in the heat. They love the heat. And they also divide by binary fission. Remember what also divides by binary fission? Mitochondria. Yes. Alright. So, we'll be moving on to bacteria. Bacteria is what? Bacteria, you know what a bacteria is, but bacteria can also benefit humans. So, for example, it can develop vitamin K, which can be used in blood clotting, and this type of bacteria is called mutualistic symbiote. So it does benefit from the human body, but also gives back something in return. It's like, let's say a bacteria is staying over at the human house, for instance, and it pays rent. You know, it benefits the human and the bacteria is also benefited. So there are other like harmful bacteria, pathogens, for instance, and they provide no benefit. The human wants them out, you know, triggers the immune system, and then he's like, boom, get out, don't stay in my house. So types of bacteria. So you got four types of bacteria. So you got obligate aerobes, which require O2, oxygen. So aerobe aerobe oxygen. Alright, obligate anaerobes, they die in the presence of oxygen. Facultative anaerobes, they switch between aerobic and anaerobic. And aerotolerant, they can't use oxygen and they aren't harmed by it, they're just neutral. So, cell walls, there are two types of cell walls. Alright, so gram positive and gram negative. Alright, so gram positive is made of peptoglycan and lipoteichic acid. Sorry, I can't pronounce that for some reason. For protection, and they maintain concentration gradient. So gram positive is made up of two things: peptoglycan and lipoteichic acid for protection and maintaining the concentration gradient. So second type is gram negative. It's a thin wall made of peptoglycan and separated by periplasmic space. So gram positive is kind of thick, gram negative is thin. Right? That's the way I want you guys to remember it. Then we got binary fission, is a way bacteria or mitochondria or archaea can reproduce, in which DNA is replicated and it's given into another copy. This process happens every 20 minutes. Bacteria has plasmids which benefit bacteria such as antibody mutation. So basically, every 20 minutes, division happens, binary fission happens. So you got one bacteria goes into two, and these two go into another two. That's every 20 minutes, by the way, and it keeps dividing, 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 dividing. All right, but of course that process in archaea and um, mitochondria could be different time-wise, but the thought is still there. Um, yep. So we were talking about plasmids, all right? So the way there's another way that, for instance, bacteria can develop new genes. They can develop new antibacterial defense mechanisms. Okay, so the first way we have it is conjugation in the male bacteria. Is the mating of the bacteria when one conjugates the bridge and transfer plasmids, genetic recombination due to F plus plasmids. So I want you to look at this diagram. All right. So the male, the male bacteria has a F plus plasmid, which means it has the bridge. So it goes to the participant, which is a female bacteria due to her F plasmid, which is F negative, and then the F plus connects a bridge, conjugates a bridge, and then transfers part of the DNA to the other participant, and then they connect, and then after that they separate, and the new donor has an F plus plasmid, and they can recombine their DNA with the new DNA they got. Moving on, we have transduction. So it's genetic recombination where virus infects bacteria and plants its DNA into the bacteria, and some DNA goes to bacteriophages. The virus carries viral DNA and the bacteria chromosome to another bacteria. So I want you to imagine this with me. So let's say we have this is a bacteria, and then we have a virus, okay, plants its DNA, all right. The virus DNA recombines with the bacteria DNA, and then the virus comes again and takes both the DNAs of the virus and the DNA of the bacteriophage or bacteria, and then it goes to another one. This is where genetic recombination happens. Um, Mix of viral DNA and bacterial chromosome. So what is the life cycle of a bacteria? We said we are going to be speaking about that. So I want you to look at here, the red dot. So bacteria starts at the log phase. A new environment, new place. The bacteria, needs to get adapted to a new environment. So after that happens, there's the exponential phase or the log phase in which the bacteria keeps increasing exponentially, binary fission, boom, 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 every 20 minutes, replicating, replicating, replicating. All right, they have all the resources in the world with them. Then they get to a certain spot, it's called the stationary phase. They stop replicating, they meet the reproduction maximum, and the reproduction is reduced, the rate is reduced, all right, and it's just boom, like resources, stuff like that, and then death occurs, which declines, all right. The amount of bacteria decline, no resources, the environment is not supporting them anymore, and yeah, that's the life cycle of a bacteria, all right. And then positive sense, a single-stranded RNA genome is translated to a functional protein by ribosomes, and negative sense, a single RNA acts as a template for the synthesis of complementary strand, which used for protein synthesis. So what is the way viruses reproduce? Basically, they hijack, replicate, and produce viral progeny. And a retrovirus, this is a bacteriophage, by the way, this is not a retrovirus. A retrovirus single strand RNA has two identical RNA and carries a transcriptase that synthesizes RNA to DNA. So remember how I spoke about um, how just the bacteriophage plants its DNA into a bacteria and then now has bacteria chromosome and the viral DNA. Yeah, that's how they do it. They hijack themselves, yeah, and then they steal other people's property. So that's it for cell theory, viruses, and bacteria, archaea. Alright, if you have any questions, please let me know. Subscribe, like, share your video, and tell me if you have any comments. Thank you so much.